Hello students, myself Bhanu Solanki, Sri Chaitanya Senior Faculty. Today we are going to discuss about one more important chapter of NEET, Biomolecule. Why I said that it is important chapter? Because this chapter every year constitute around 3 to 5 questions of your paper of NEET. So it is very important chapter and have a lot of informations useful for NEET as well as for your upcoming career in as a medical student also. So let's start this chapter, Biomolecule. There is a wide diversity in the living organisms in our biosphere, and now a question that arises in our mind is that all living organisms are made up of the same chemicals. Biochemical organization of all kind of organisms is same. The difference is only in the relative proportion of that. As mentioned, elemental and the compounds, both type of chemicals present into the cell in the different proportions into the different amounts. In this chapter, when you compare a sample of the earth crust and also a sample of the human tissue, you can see that what is the scenario. Look, a comparison of the element present in non-living, yani earth crust, and in the living matter is mentioned here. Hydrogens are 0.14% in earth crust while they are 9.5% in human body. I want to tell you that this value was earlier given different in different versions of NCERT. This is the latest version, 9.5. Carbons 0.03% while 18.5% in human body. Oxygen 46.6% in earth crust while 65% in human body. Nitrogen very little, 3.3%. Sulfur 0.03%, 0.3% there. Sodium, calcium, magnesium and silicon. A brief comparison is given here. I suggest memorize this table by heart. This is one of the probable question. Either in terms of amount they can ask or in terms of arrangement, in ascending arrangement or descending arrangement. Type of questions are very frequent and possible from this table. How to analyze chemical compositions? So to do this, there is a acid titration method. By using trichloroacetic acid, we can carry out this analysis. And when we carried out this analysis, you remember we will crush plant or animal tissue in trichloroacetic acid and then pass it through the cheesecloth or cotton. Then the part which remain left over the cotton or the cheesecloth, that is called acid insoluble pool. And the part which pass through it, that is called acid soluble fraction. Acid insoluble fraction means the part which is in cotton cheese. One called filtrate or technically acid soluble pool. So when we compare it, you must remember all kind of polymers, means proteins, polysaccharides, nucleic acids are present in acid insoluble fraction, and lipids are also present in acid insoluble fraction, while monosaccharides, disaccharides, amino acids, nucleotides, they all are present in acid soluble pool. In other words, you can say that 3 types of macromolecules, protein, polysaccharide and nucleic acid, and lipids are present in acid insoluble pool, while all kind of monosaccharides, disaccharides, amino acids, nucleotides are present in acid soluble pool. Lipids are not a macromolecule. Even after that, they are present in acid insoluble pool. Why? Because lipids are generally present in membranes, and when we will homogenize a membrane, so it will break into the tiny vesicular structures, and these vesicles cannot pass through the cotton or the cheesecloth. That is why they retain within the acid insoluble fraction. Here there in this paragraph they are mentioning two things. All the carbon compounds that we get from the living tissues are called biomolecules. Definition: all the carbon compounds that we get from the living tissues can be called as biomolecules. Second thing, dry weight and fresh weight. When we take a plant leaf and weight it, it is called fresh weight. With help of oven, when we dry it and again we take its weight, that is called dry weight. Here there, ash analysis can also be carried out. Elemental analysis gives element composition of the living tissues in form of hydrogen, oxygen, chlorine, etc. While analysis for compounds gives an idea of the kind of organic substances. Elemental analysis about the inorganic elemental composition, and analysis for compounds for the organic compounds. Later on, in living tissues, from a chemistry point of view, one can identify functional groups like aldehyde, ketone, aromatic, etc. But from the biologist point of view, we shall classify them into the amino acids, nucleotides, fatty acids, etc. Amino acids are the organic compound containing an amino group, an acidic group, as substitutes on the same carbon, that is the alpha carbon. Hence, they are alpha amino acid. Protein forming amino acids taking part in cell protein synthesis phenomena are alpha amino acid. What they are? They are substituted methane, because all four valencies of the carbon occupied by the four different groups. There are four substituents group occupying to the four valencies. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types, means 20 types of amino acids forming to the protein. R group of these proteinaceous amino acids may be in form of hydrogen, or it may be in form of methyl group, or it may be in form of hydroxymethyl group, or it may be other. When R is in form of hydrogen, example is glycine, you can see in glycine R is in form of H, in alanine R is in form of CH3, in serine it is in form of hydroxymethyl group. So R is variable, accordingly amino acid is also variable. The chemical and physical properties of the amino acids are essentially of, very important paragraph, the chemical and physical properties of the amino acids are essentially of the amino group, carboxyl group and the R group. Based on the number of amino group and carboxyl group, amino acids may be acidic, may be alkaline or may be neutral. Here examples are given that the acidic amino acids means glutamic acid, basic means lysine and neutral means valine. So valine is a neutral amino acid, lysine, arginine, histidine, all three are basic or alkaline amino acids. Glutamic acid, aspartic acid, they are acidic amino acids. One more thing, here there, aromatic amino acids, examples of aromatic amino acid is also given here there, including tyrosine, phenylalanine and tryptophan. A particular property of the amino acid is the ionizable nature of NH2 and COH group. NH2 would be ionized as a NH3 plus and COH group would be ionized as COO minus. When both amino group is ionized and carboxylic group is also ionized. So this state of the amino acid having both positive charge and negative charge is known as deuterion. At any particular value of the pH, amino acid may exist as a deuterion. From this pH, as we increase pH or we decrease pH in both of the direction, the net charge over the amino acid would be either positive or negative. Means it would not be a deuterion. In the deuterion state, net charge is 0. Then after amino acids, lipids are given there. They are water insoluble. They could be simple fatty acids. A fatty acid is a carboxyl group containing a long chain hydrocarbon substances. This R group may be a simply methyl group or a ethyl group or higher number of CH2 groups, vary from 1 carbon to 19 carbon. Palmitic acid means 16 carbon including carboxylic acid. Arachidonic acid 20 carbon including carboxylic group. Palmitic acid 16 carbon containing fatty acid. Stearic acid 18 carbon containing fatty acid. Arachidic acid as well as arachidonic acid, both are different. Arachidic is a saturated fatty acid, arachidonic is the unsaturated. Both have 20 carbons. Fatty acids could be saturated or unsaturated, as I said. If double bonds are present, so they are unsaturated. If not present, they are saturated. Another simple lipid is the glycerol, mentioned here, which is a trihydroxypropane, means CH2OH, CHOH, CH2OH. Many lipids have both glycerol and fatty acids. When glycerol get esterified along with the fatty acids, this structure is known as glyceride. They can be monoglyceride if one hydroxyl group esterified with one fatty acid. If two hydroxyl group esterified with two fatty acid, diglyceride. If three, then it is a triglyceride. So one glycerol plus three fatty acid resultant triglyceride. Yes. One more difference is here, that is mentioned fats and oils. Lipids can be classified as fats and oils based on the melting point. Oils have lower melting point, hence remain as oil in winters. Some lipids have additional group phosphorus and they are known as phospholipids. You know, we have discussed in cell membrane that phospholipid is the representative of the lipids in plasma membrane. Lecithin is one of the example of phospholipid. Let me show you in diagram. This is palmitic acid, CH3, CH2, whole 14, COOH. So 1 plus 14 plus 1, yani 16 carbon. This is the structure of glycerol, CH2OH, CHOH, CH2OH. When over the one glycerol, this part I highlight, this is glycerol. One fatty acid, second fatty acid and third fatty acid get attached, so what it become? Triglyceride. One of the phospholipid is mentioned here there, in which one glycerol, two fatty acid, one phosphoric acid and one nitrogen containing group choline is present. So this structure is known as lecithin. Another lipid molecule is mentioned here as a cholesterol. We know it is a steroidal chemicals. Cyclopentanoperhydrophenantine nucleus structure they are. So these structures given in this figure 9.1 you have to keep memorized by heart. Come back. Living organisms have a number of carbon compounds in which heterocyclic rings are present. So we are talking about the purines and pyrimidines actually, means nitrogen bases, adenine, guanine, cytosine, uracil, thymine. They all have the heterocyclic ring. If a phosphate group is found esterified to the sugar, they are called nucleotides, means nitrogen base plus sugar, that is nucleoside. And over the nucleoside phosphoric acid get attached, so what it become? Nucleotide. And nucleotides are the building blocks of, are the monomers of nucleic acids. So adenine with sugar become adenosine, guanine with sugar become guanosine, cytosine with sugar cytidine, uracil with sugar uridine, and thymine with sugar thymidine. So adenosine, guanosine, cytosine, cytidine, thymidine and uridine, they are nucleosides. Now if phosphate attach with them, adenylic acid, guanylic acid, cytidilic acid, thymidilic acid, uridylic acid, they all are nucleotides. See from the image. This is adenine. Now on the sugar's first carbon, if adenine attach, so it become adenosine, means nucleoside. And on the fifth carbon as phosphoric acid attached, so what it become? Adenylic acid. So nitrogen base, nucleoside, nucleotide. Similarly, uracil, nitrogen base, attached at the first carbon of sugar, so it become uridine, means you understand nucleoside. And if on fifth carbon, if I attach what? Phosphoric acid, so what it become? Uridylic acid, means nucleotide. So nucleic acids like DNA and RNA consist of the nucleotides. DNA and RNA function as a genetic material in different organisms. Then primary and secondary metabolite. This also is very important part. Here there are only few things that you have to keep in your memory. These are the primary metabolites. What? Carbohydrate, lipid, proteins, etc. However, when we analyze plant cells, fungal cells and microbes, one would found thousands of compounds other than the primary metabolite, and they are known as secondary metabolites, including alkaloids, flavonoids, rubber, essential oils, antibiotics, coloring pigments, scents, gums, spices, etc. So what is the point? That secondary metabolites generally present in plant, fungus and microbial cell. This table we have to keep in memory by heart. Pigments including carotenoid and anthocyanin, alkaloids, morphine and codeine, terpenoids, monoterpenes and diterpenes, essential oils, lemongrass oil, etc. Toxins, abrin and ricin, lectins, concanavalin A, drugs, vinblastine, curcumin, etc. and polymeric substances, rubber, gum, cellulose. This table with the categories and example we have to keep in memory. What is the definition of secondary metabolite? Here it is mentioned. That primary metabolites have the identifiable function and a known role in normal physiological processes. So it means those substances which do not have identifiable function and which do not play any known role in physiological processes, they are considered to be the secondary metabolite. Secondary metabolites have ecological importance. This is the one of the important point. Means they provide protection against the grazing, etc. Then there is biomacromolecules. The definition of biomacromolecule mentioned here: any compound that have molecular weight ranging from 18 to 800 Dalton are known as micromolecules. And they are present in acid soluble fraction. In the acid soluble pool they are present. Means all those compounds that has molecular weight ranging from 18 to 800 Dalton are part of acid soluble pool, and those which has molecular weight more than 800 Dalton, they are part of acid insoluble pool. So here they mention that molecules, these classes of the compounds with the exception of lipids, have molecular weight in range of 10,000 Dalton. Means polysaccharides, proteins and nucleic acids which are part of acid insoluble pool, they have molecular weight more than 10,000 Daltons always, yani more than 800 Daltons, so they are macromolecule. But lipid is also present in acid insoluble pool, but it is not a macromolecule. I have already told you that why it was present in acid insoluble pool. That's it. So the molecule in the insoluble fraction with the exception of lipids are polymeric substances. Then why do lipids whose molecular weight is not exceeding to the 800 Dalton comes under the acid insoluble fraction? We have already discussed. The acid soluble pool this line also represent roughly to the cytoplasmic composition. Second, macromolecules form cytoplasm and there's the organelles. The macromolecules from the cytoplasm and organelle become the acid insoluble fraction. This line you have to keep in your memory. I repeat again, acid soluble pool represent roughly to the cytoplasmic composition, and the macromolecule from the cytoplasm as well as organelle become the acid insoluble pool. Here the values are given: water 70 to 90%, proteins 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acids 5 to 7%, and ions 1%. Means again ascending, descending order arrangement of various chemical present in cellular mass table and their values. First of all, they mention about the proteins. Proteins are polypeptides. Proteins are linear chains of amino acids linked by peptide bond. Proteins are polypeptides. Proteins are linear polymers, and in the proteins, the amino acids held together by peptide bond. One more point. Proteins are heteropolymers, means the units which are taking part in protein formation are not identical. They are different types of 20 different types of amino acid. So they are considered to be heteropolymers. Each protein is a polymer of amino acids. Around 20 types of amino acids take part in protein formation. A homopolymer has only one type of monomer repeating units. This information about the amino acid content is important as later in your nutrition lesson also. Some of the functions and examples of proteins are given here. Collagen, it is an intercellular ground substance of the animal cells. Trypsin, it is an enzyme. Insulin is the hormone. Antibodies are the fighting infectious agents. Receptor includes sensory receptions. GLUT4, it is a kind of a protein present in plasma membrane that enables transport of the glucose across the cell. These examples, particularly GLUT4 and insulin, are very important. Collagen is the most abundant animal protein. Most abundant animal protein. While rubisco, ribulose bisphosphate carboxylase oxygenase protein is the most abundant protein into the whole biosphere. You know why? Because entire planet is running because of the photosynthetic product, and that photosynthetic product is contributed by rubisco. Then polysaccharides. The acid insoluble pool beside polypeptides have polysaccharides, yani carbohydrates, as another class of the macromolecules. Polysaccharides are long chains of sugars, yani sugars are the monomer. They are thread-like structures containing different monosaccharides as a building block. Means unlike proteins, carbohydrate may be homopolysaccharide or may be heteropolysaccharide. If any polysaccharide made up of same identical units, so it is called homopolysaccharide. Starch, glycogen, cellulose, chitin, they all are homopolysaccharides. Cellulose is a polymeric polysaccharide consisting only of one type of monosaccharide, that is the glucose. Cellulose is a homopolymer, as I mentioned already. Starch is a variant of this but present as a store food house. Means starch and glycogen, they are storage polysaccharides, while chitin and cellulose both are structural polysaccharides. Means they meant for structure formations, while glycogen and starch meant for storage purpose. So starch is the another variant. Animals have another variant that is called glycogen. So glycogen and starch are equivalent. Inulin is a polymer of, very important point, inulin is a polymer of fructose. Glucose, starch was a polymer of glucose. Glycogen was polymer of glucose. Cellulose was polymer of glucose. Chitin was polymer of N-acetyl glucosamine. And inulin is a polymer of fructose. In any polysaccharide chain, the right end is called reducing end and the left end is called the non-reducing end. Suppose this is a polymer. So this is the right end and this is the left end. Right end is the reducing end and left end is the non-reducing end. At its branches as shown into the form of cartoon figure 9.2, starch forms a helical secondary structure. Means in starch there are two components, amylose and amylopectin, and in this amylopectin part we see some branches, helical structures, that is the helical structures, and because of the helical structure intramolecular hydrogen bonding happen, so it attain a secondary structure. So starch has a secondary structure. Cellulose does not contain complex helices, hence cannot hold iodine. But starch as have the helical structure, so when you treat starch with iodine, this iodine will be held in these helices and it will give the characteristic blue color. So starch forms helical secondary structure. Starch can hold iodine molecule in the helical portion, that is why it appear blue colored in stem. Plant cell walls are made up of cellulose. Papers are also made up of cellulose, that we have discussed. It is a more complex polysaccharide in nature. Beside this, one more is mentioned here, that have building blocks amino sugar containing chemical modified sugars like N-acetyl glucosamine. Exoskeleton of the arthropods, for example, have a complex polysaccharide called chitin. These chitin are the complex polysaccharide and are also homopolymers, and they are made up of N-acetyl glucosamine. This is the structure of diagrammatic presentation of a glycogen. You are watching glucose, 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 glucose. This bond between two glucose units is the 1,4-glycosidic bond, and this branching point have 1,6-glycosidic bond. So in a linear form, only one type of bond, that is 1,4-glycosidic bond, and whenever branching happen, so at the branching point there would be the 1,6 bond. So cellulose is a unbranched, it means it has only 1,4-glycosidic bond, but starch and glycogen both are branched homopolysaccharide, it means they have two types of bond, 1,4-glycosidic bond as well as 1,6-glycosidic bond. Nucleic acids. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acids. These are polynucleotides, and we know that in any nucleic acid 4 types of nucleotide, either adenylic acid, guanylic acid, cytidyllic acid, thymidyllic acid. So it would be a DNA. Adenylic acid, guanylic acid, cytidyllic acid and uridylic acid. So it is a RNA. Means 4 types of nucleotides are the constituent of nucleic acids. So nucleic acids are also heteropolymers. So they are macromolecule found in insoluble fraction. These are polynucleotides. Together with the polysaccharides and polypeptides, these comprise to the true macromolecular fraction. Together with polysaccharides and polypeptides, these comprises the true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. Nucleotides are monomers. Nucleic acids are polymers. A nucleotide has 3 chemical distinct components. I already told you. Heterocyclic compound, yani nitrogen base. Second is the monosaccharide, ribose or deoxyribose, and third is the phosphoric acid or the phosphate. When sugar and nitrogen base fuse together by glycosidic bond, nucleoside form. Over nucleoside sugar, if phosphoric acid attaches at the fifth carbon, nucleotide form. You notice in figure 9.1, heterocyclic compound in the nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, thymine. Adenine and guanines are substituted purines. Two types of nitrogen bases are there, purines and pyrimidine. Adenine and guanine both are the purines, substituted purines. While the rest are substituted pyrimidines. The skeletal heterocyclic ring is called purine and pyrimidines respectively. The sugar found in polynucleotide is either ribose if it is RNA and deoxyribose if it is a DNA. A nucleic acid containing deoxyribose is called DNA and the another one RNA. Protein's structure. Protein, as mentioned earlier, are heteropolymers, we have discussed, containing strings of the amino acids. Structure of the molecule means different things into the different context. For from biology point of view, if we will talk, so we will follow biologists describe protein structure at the four level. Primary structure, secondary structure, tertiary structure and quaternary structure. The sequence of amino acid, very important line. That means the positional information in the protein. Yani what is the first amino acid, what is the second, what is the third, what is the fourth. Positional information of the amino acids within the protein is known as primary protein structure, primary configuration. Primary configuration of protein means positional information in a protein, means sequence of amino acid, simple. In a primary configuration of protein, only one type of bond is present, that is peptide bond. No other additional bonds are present in primary structure. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. I can show likewise. Amino acid, 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 amino acid. Suppose I start to count from here onwards. So this is the n, this is the n plus 1, this is the n plus 2, this is the n plus 3, likewise. This is first amino acid, this is last amino acid. Of this amino acid, N group is free, so this is called N-termine. And from this amino acid, carboxylic acid group is free, so this is called C-termine. So in any polypeptide, in any primary protein, one end is a N-terminal and one end is a C-terminal. Which is the first amino acid, which is the second, and so on. It is called primary structure. A protein is imagined, we have discussed. So here it is shown one amino acid, 1, 1, 1, 1, 1. So this end would be the N-termine, as I said earlier, and this is the C-termine. The first amino acid is called N-terminal amino acid, and the last amino acid called C-terminal amino acid. A protein thread does not exist throughout as an extended rod. A protein cannot to be present in their entire length in form of a thread-like structure. It will coil, it will twist around it, and as it coil, condense, twist, then it will attain different configurations. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in form of alpha helix, similar to a revolving staircase. Of course, only some portion of the protein thread are arranged in form of a helix. You see, what does the mean of it? Suppose this is a protein, at some point it become alpha helix and then again. So up to here what it is? Primary. What it is? Primary. But in this region what it attain? Alpha helix. This is the structure of alpha helix. So alpha helix is a kind of secondary structure. Alpha helix in protein are always right-handed. Alpha helices are always right-handed structures. Other regions of the protein thread are folded in form of the other forms in what it is called secondary structure. This is the secondary structure called beta pleated structure. This is another secondary structure called alpha helix structure. When alpha helix form or beta pleated structure form, due to this coiling, due to this folding, another additional bond established, that is hydrogen bond. So in an alpha helix there are two types of bond, peptide bond and hydrogen bond. In beta pleated structure also there are two types of bonds, peptide bond and hydrogen bond. So in secondary structure what is the additional bond? Hydrogen bond. Secondary structure is of two types, alpha helix and beta pleated. In alpha helix you must remember the helix would be always right-handed. Other reason, that's it. In addition to the long protein chains also folded up to upon itself like a hollow woolen ball, giving rise to the tertiary structure. Look this diagram. That if another turn of the coiling and folding happen, so it become like a ball of the cotton or the wool. That structure attains some more additional bonds. It may be disulfide bond, it may be electrostatic interactions, it may be hydrophobic attractions, etc. When any protein beside peptide bond and hydrogen bond attain these type of bonds also, so it become a three-dimensional granular structure, and this configuration called tertiary structure. This tertiary structure give us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities. Very important point. You know why? Because you must remember that majority of enzymes which are made up of protein can act as an enzyme when they are in a tertiary form. Yani most of the protein enzyme work in a tertiary form. That is why they are absolutely necessary for the biological activities. Primary, secondary, tertiary. Whenever any protein has molecular weight on a higher side, more than 100 Dalton, so in that condition, you know what happen? More than one polypeptide chains participate. When more than one polypeptide chains participate, so in what geometric manner, in what spatial manner they remain arranged, that arrangement, that geometric arrangement is known as quaternary structure. Some proteins are assembled of many more than one polypeptide subunits. The manner in which the individual folded proteins are arranged with respect to the each other, yani linear string of the spheres or spheres arranged one above another in form of a cube or plate, in the architecture of a protein, otherwise called quaternary structure of a protein. So quaternary structure means you can assume geometrical arrangement of the polypeptide subunits. Simple. Means quaternary, for the quaternary structure, the protein must be made up of more than one polypeptide chain. A single polypeptide chain containing protein can never attain quaternary structure. To attain quaternary structure, number of participating polypeptide subunits must be more than one. Then only one polypeptide unit can arrange in a specific manner with respect to the another, and that spatial arrangement is known as quaternary structure. For example, in hemoglobin, as we know that there are 2 alpha chains and 2 beta chains. So these 4 polypeptide chain, 1 alpha, 1 beta, 1 alpha, 1 beta, remain arranged in what manner with respect to each other? That is the quaternary configuration. Another point to be discussed in this chapter is the enzyme. Remember, almost all enzymes author is not saying all enzymes are protein. Author is saying almost all enzymes are proteins, because there are some exceptions. As you know, ribozymes are protein. There are some nucleic acids that can act as an enzyme, and we know what they are, ribozymes. So except ribozymes, all enzymes are protein. One can depict an enzyme by a line diagram, यानी कि sequence of amino acids. An enzyme, like any protein, has the primary structure, that is the amino acid sequence. Then an enzyme, like other protein, has the secondary and tertiary structures also. When you look at tertiary structure upon itself, the chain crisscrosses itself and make various kind of bonds, and then they attain to a different structure. Enzyme catalyze different inorganic catalyst in many ways, but the major difference between catalyst and enzyme, one of the major difference needs to be mentioned, and that is that at high catalyst, inorganic catalyst can work at the high temperature and high pressure. At the high temperature and high pressure, while enzyme get damaged at the high temperature and pressure. Means one of the fundamental difference between catalyst and enzyme is what? That enzymes are temperature, pH and pressure sensitive. They cannot tolerate extremities or fluctuations of temperature, pressure and pH. While catalysts are extremes, they can work on extreme of temperature, extreme of pH, extreme of pressure. Enzymes, however, generally work in an optimum range. However, some enzymes isolated from the organisms who normally live under the extreme conditions, they can work even up to the 80 to 90 degrees centigrade. But this is not a normal range of enzyme working. In general, enzymes tend to work below 40 degrees centigrade temperature. How do enzyme, here, there, then again, chemical reactions part is given. It is a very simple paragraph. Here, there, nothing to be worried, just only few points you have to keep in your mind, that it is a general rule of thumb that in the that the rate of reaction on temperature variation by the temperature among the catalyzed reactions proceed very fast, higher than the uncatalyzed reaction. Variation of 10 degrees centigrade temperature lead to the doubling of the rate of reaction. Here this reaction, as mentioned, carbon dioxide plus water in the presence of carbonic anhydrase convert into the carbonic acid. This reaction happen with help of enzyme, and this reaction also happen without an enzyme. But as you are watching the arrow, when this reaction happen in absence of enzyme, then the rate of reaction would be very slow, means the number of product molecules would produce in a very less amount. That is why author is saying that in a biological world, all enzymes are enzyme catalyzed reactions. Here, there, in the absence of an enzyme, this reaction is very slow with about 200 molecules of carbonic acid being formed in an hour. However, by using the enzyme present within the cytoplasm, the rate of reaction is extremely tremendous, and around 60,000 molecules of the product will form every second. It is carbonic anhydrase is one of the fastest enzyme, and this is called turnover number. 6 lakh molecules of the substrate get transformed into the product per second, means 36 lakh substrate molecules per hour. Here, there, this is also about the enzyme's role. Now, how do enzyme perform to the chemical reaction is going to be discussed. that the substrate convert into the product. And when substrate convert into the product, this reaction made possible with the help of the enzyme. How? Enzymes has some cavities or pocket-like structures on its surface, and they are known as active sites. Overactive site enzyme attach and it lead to the work. This graph is very important. It is indicating what? That to convert into a product, when substrate is to be converted into a product, first of all, substrate have to attain one of the unstable state. This one unstable state of the substrate is known as transition state. Because when it is in an unstable state, then only chemical bonds of the substrate can break and new bonds can break. So the amount of potential energy which is required to create unstable transition state of the substrate is known as activation energy. Enzymes do what? Enzymes simply do what? They reduce the value of activation energy. Means now substrate can easily achieve their unstable state, means transition state, and get easily converted to the product. So there are two graphs. First graph shown in red color indicating this much of activation energy. Second graph is showing a relatively less amount of activation energy. Means what enzymes are doing? Enzymes are decreasing to the value of activation energy so that substrate can easily achieve their transition state and can convert into product. When substrate convert into product, maybe one or more such kind of a transition states or unstable states can appear. So here this is the presentation that enzymes comes in contact with the substrate, then the enzyme substrate complex will form. This enzyme substrate complex then turn into the temporary enzyme product complex, and that enzyme product complex then separate. Enzyme and product become separate from each other. Catalytic cycle is shown here. First the substrate bind to the active site of enzyme, then binding of the substrate on active site lead to the change in the active site so that it can firmly attach or bind to the substrate. The binding of substrate induces enzyme to alter its shape. Shape of substrate, shape of enzyme, so that fitting of the substrate become more tight around the substrate. The active site of the enzyme now in close proximity of the substrate, closest proximity, so that the bond making and breaking can more possible. The enzyme release the product, and now this enzyme is free to perform the another reaction. This is the catalytic cycle. It is also important because sequence based question here also made possible. Then what are the factors that can affect to the enzymes? Yes, temperature, pH, concentration of the substrate, etc. I already told you that the enzymes are sensitive towards the temperature, pH and pressure. Enzymes can work only within the optimum range of temperature. When temperature range is optimum, as shown here, the rate of reaction would be highest. But from the optimum temperature, as we decrease temperature or increase temperature, in both of the direction, rate of reaction will decrease. Same is the effect of pH. At the optimum pH, the rate of reaction would be highest. At the optimum pH, the rate of reaction would be highest. But as from the optimum pH, we decrease or we increase pH, rate of reaction will decrease. This is effect of temperature and pH. So enzymes generally function in a narrow range of temperature and pH. Each enzyme show its highest activity at a particular temperature and pH, and that is known as optimum temperature and optimum pH. Activity declines both above and below, we have discussed. Next factor is a concentration of substrate. See in the diagram. This is the concentration of substrate. So when we plot a graph in between velocity of enzyme catalyzed reaction and substrate, what we are watching, that the rate of reaction is directly proportional to the substrate concentration. rate of reaction is directly proportional to the substrate concentration. The particular value of substrate upon which an enzyme achieve half of its Vmax, that concentration of substrate upon which an enzyme achieve half of its Vmax, that concentration of substrate called Michaelis-Menten constant or Km value. For an ideal enzyme, its Km value should be low. When with the increase in substrate concentration, velocity of the enzyme catalyzed reaction first increases, the reaction ultimately reaches to the maximum velocity and then become constant, that is the Vmax. This is because the enzyme molecules are fewer than the substrate molecules, so all enzymes as get saturated. Afterwards, there is no further chance of the increment of the rate of reaction. The activity of enzyme is also sensitive to the process of specific inhibitor chemicals. Here the process of the shutting down of the enzyme action is known as inhibition. Some of the competitive inhibitors they are mentioning here. If the shape of a chemical is similar to the shape of a substrate, so this chemical and substrate will compete to achieve the active site. This substrate we are calling substrate, but this chemical which is structurally resembling to the substrate, we started to call it substrate analog. The competition is in between substrate and substrate analog to achieve what? To active site. There are some examples mentioned. Succinate dehydrogenase enzyme. Substrate is a succinic acid, but structure of malonic acid is resembling to the succinic acid. It means malonic acid doing a competitive inhibition of succinate dehydrogenase. One more example is here mentioned, that the various kind of drugs that we use to control the pathogens, they are also working at the principle of competitive inhibition. I repeat, in a competitive inhibition, substrate analog compete with substrate, not with enzyme. Enzyme is the objective. Substrate want to attach on enzyme, substrate analog also want to attach on enzyme. So competition is going on between substrate and substrate analog. Two examples: inhibition of succinate dehydrogenase by malonic acid. While attempting a question, be careful. Malic acid nahi, malonic acid should be there. So these are the factors. Then enzyme get classified into the 6 classes by International Union of Biochemistry. First is a oxidoreductase, also called dehydrogenase. Means what they are doing? They catalyze oxidoreduction reactions. Means suppose substrate is reduced, another substrate is oxidized. So substrate reduced get oxidized and the substrate reduced, oxidized get reduced. Simple. Second is a transferases. What they called? They group transfer. But this group should not be hydrogen, because if this group which is going to be take part in transfer, if hydrogen, so it is a oxidation reduction. So transfer of group but other than the hydrogen. Third is a hydrolases. What they do? They catalyze hydrolysis of the ester bond, ether bond, peptide bond, glycosidic bond, CC bond, carbon halide bond, PN bond, etc. Lyasase. They catalyze removal of the group from the substrates by mechanism other than the hydrolysis. And what they do? They leave double bond into the product. See, product become XY and another product C double bond C, means double bond established. Isomerases, we know very well, includes all enzymes that catalyze the interconversion, either optical interconversion or geometric interconversion or positional interconversion. Isomerases. Ligases, we have discussed in the DNA replication. These enzymes catalyze the linking of together of the two compounds, carbon oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen, etc. Then cofactors. Enzymes are composed of the one or several polypeptide chains. However, there are number of cases in which non-protein part, also called cofactors, are bound to the enzymes to make them catalytically active. Yani what is the function of cofactors? Catalytic activation of enzyme. In these instances, protein portion called apoenzyme. Protein part is known as apoenzyme, and non-protein part we have discussed also called cofactor. There are some enzymes in which beside protein part, non-protein part is also involved. This protein part is known as apoenzyme and non-protein part is known as cofactor. These cofactors are of three types: prosthetic group, coenzyme and metal ion. What are the prosthetic group? Prosthetic groups are organic compounds and are distinguished from the other cofactor that their binding with the apoenzyme is very tight. So whenever any cofactor attaches with apoenzyme firmly, tightly, so this is called prosthetic group. If it join with apoenzyme loosely, so it is called coenzyme. Or if it is an inorganic and joining is a loose, so it is called metal ion. Yani metal ions are inorganic, coenzymes are organic, both attach with apoenzyme in a loose manner. Prosthetic group are mostly organic, but it may also be inorganic. But their binding with the apoenzyme would be tight and firm. So prosthetic group on one side, coenzyme and metal ion on another side. Binding firm, binding loose, may be organic, may be inorganic. Coenzymes are always organic, metal ions are always inorganic. Here the examples mentioned. For the peroxidase enzyme and catalase enzyme that lead to the breakdown of hydrogen peroxide to the water and oxygen, heme is the prosthetic group. In the catalase enzyme as well as in peroxidase enzyme, heme is the prosthetic group. Coenzymes are organic compounds, but their association with the apoenzyme is loose transient. Coenzymes serve as a cofactor in number of different enzymes. Examples are mentioned. NAD, NADP, both are coenzymes. NAD and NADP, yani coenzymes, are mostly of vitamin origin, means they are made up of vitamins. NAD and NADP both are made up of niacin, that is a vitamin. Metal ions, they are always inorganic and they form coordinated bond. What they form? Coordinated bond with the side chains of the active site at the same time, one or more coordination bond with the substrate. Means active site, metal ion and substrate. Active site, metal ion, substrate. Metal ion will form coordinated bond with active site and on other side they make coordinated bond with substrate. Means metal ion connect to the substrate and apoenzyme with the help of coordinated bonds. Example is mentioned. Zinc, it is a metal ion yani cofactor for the proteolytic enzymes carboxypeptidase. This paragraph is very important from exam point of view. Almost every year one question arises from these three paragraphs. So from biomolecules chapter you cannot avoid this paragraph. Read it frequently whenever you get time. So this is all about the biomolecule and various questions that have already been asked from this chapter. Already you are aware about them. Once go through them and read this chapter with this specification. It would help you to crack the questions so that you can pursue your goal. Thank you very much. All the best.